All right, welcome to the more advanced genetics overview. In this overview, we'll be taking a look at one of Morgan's experiments. Morgan did a lot of experimentation with fruit flies, and we'll take a look at one of his experiments that actually placed a gene for the first time on a chromosome. Scientists for a very long time hypothesized that genes were on chromosomes. They thought they were there, but nobody had done an experiment to definitively put a gene on a chromosome. And then we'll take a look at something called sex-linked inheritance patterns that we see when alleles are on the X chromosome. Remember, males have one X and one Y, and females have two X chromosomes. That also leads to a difference in inheritance as a result of something called X inactivation in female mammals. One of the X chromosomes, one of the two, will inactivate, kind of condense down to a nubbin of nothing in each of the female mammal body cells. So we'll take a look at that process. And then finally, alteration in chromosome structure and number. These are usually mistakes that occur during meiosis in the chromosomes, and that can result in problems in the offspring. All right, so first, Morgan's experiment, work with Drosophila. He did this because they really bred at a very high rate and get a new generation every 2 weeks, so you can take a look at the characteristics of the offspring very quickly. And the genetics were very simple, just 4 pairs of chromosomes, and we have our male and our female shown here. Now, Morgan did things a little differently as far as his characteristics with his Drosophila. He took a lot of liberties because he was the first person to really seriously use Drosophila as a model organism for genetic studies. People thought he was crazy, and he got a lot of flack for working with Drosophila, but today it is the #1 used model organism for genetic studies, because it is just so very, very useful and simple to work with. So he named his characteristics differently than Mendel. He, instead of having dominant versus recessive, he had something called wild type as opposed to mutant. So wild type is the one you'd normally see in the wild, the most common type of allele. Okay, so wild type means kind of like dominant in Mendel's naming system, and mutant would be the less common. Not that it's anything wrong with it necessarily, but it's the less common, so more like the recessive. So a wild type eye in a Drosophila, if you've ever looked at one pretty close, these are the fruit flies that like to swarm your bananas, would be a red eye. And a mutant, less common, would be a white-colored eye. Now, this is a really big overview of Morgan's experiment that placed a gene on a chromosome, and the details are located in the chapter, so please read over the experiment prior to listening to this just quick review of the experiment. But as you can see, he follows the typical Mendelian experiment, my friends. True breeders. And when he looked at the crossing between the male and the female here in the parental generation, he found that the F1 generation only had red-eyed organisms, and the F2 had the 3-to-1. But every time he did this experiment, only the males would have white eyes. There would never be females with white eyes. Never. And that was really strange because white-eyed females do exist. They do exist. It's not that they don't exist. So he thought to himself, what is the only difference in terms of chromosomes between males and females? The X and the Y. So just like in humans, males have an X and a Y chromosome, and females have 2 X chromosomes. It must be something with the sex chromosomes, the X and the Y. And he thought to himself, now, okay, again, there are white-eyed females, So it shouldn't be on the Y chromosome, because if it was on the Y chromosome, this allele for white eye color, we wouldn't have white-eyed females. So it must be on the X. So his naming system, or his notation system for his alleles is a little different. He always named it after the mutant, so white, W for white eyes, and it's always lowercase for both wild type and mutant alleles. And if it's a wild type, it gets a little plus superscript, and if it's a mutant, it gets nothing, nothing for that allele. So we have wild type alleles. So this is a true breeder, female red eye in this experiment, and we have a wild type allele, and we have the male, the Y with no allele, and the X with the mutant allele, the white-eyed allele. And we get two types of sperm, Y sperm and X allele with the white-eyed allele, mutant allele. And we only get one type of egg over here from this lady because she is a true breeder and she only produces the wild type allele. So here is the F1 generation. We have red-eyed females and white-eyed males. Okay? So we have one of each here, and wild type, just as in Mendel's system, wild type being similar to dominant and mutant being similar to recessive. Wild type dominates over wild type dominates over mutant if we have one of each. So this female will be red eye. And we don't have any allele for eye color on the Y, so this male is automatically red eye, and that matches up over there with what he saw. And then we take, to get our F2, we take this male and we mate it with that female. So we have her egg possibilities here, his sperm possibilities here. And when we do the genetics and we look at the genotypes, we find that the only possible way to get a white-eyed fly is if he's male. He put a gene for eye color on the X chromosome. He did it. Congratulations, Morgan. Good job. Okay. Now, sex-linked inheritance, as we've seen, we have a unique inheritance pattern where we see more often times that males will have certain characteristics as opposed to females. So, and that in the human population, that has a lot to do with the fact that the X chromosome is so much larger than the Y chromosome. So the X chromosome, as you can see here, huge in comparison. So we have a lot more genes on the X chromosome as compared to the Y chromosome. And when you have this kind of inheritance pattern in a family where we have carriers of a particular trait we're looking at. So for instance, the females are carrying one copy of the recessive allele for hemophilia, and but they have a good copy as well, so they don't have hemophilia. But all the females do have at least one copy, and then this mother has a child with this father, and this boy is actually a boy, there's a square. He is a hemophiliac because he inherited the diseased X copy of the allele from mom, and he doesn't have another X possibility from dad, so he will more often, or males more often, will have these sex-linked diseases or traits compared to females. So here we have an example, and the notation here is a little different for the X-linked. We always have the sex chromosome shown, so for the female X and the male X and Y, and the allele is a superscript. If it's a normal allele, and we're talking about human characteristics, we give it a capital A, and if it's a diseased allele, it is a lowercase. Now we can see the result of this Punnett square shown here, and we will pick this back up in the next video and continue on with talking about some of the differences between males and females in terms of inheritance patterns. So again, we're going to stop for now, and we're going to pick